Pathogenic white vascular clot formation triggered by host enzymatic furin cleavage in the viral spike protein. Refer to the previously posted intravascular amyloid white clot deposition presentation. Pause each illustration for further detailed study. Furin cleavage structure. Protease is an enzyme that breaks down proteins and peptides. It is activated by substrate presentation in humans and animals, encoded by this gene. Raw anomalous intravascular casts samples from a single donor. Representative anomalous intravascular casts, AIC clots, collected during routine postmortem care. Raw clot samples. Furin is a serine protease that is a convertase host cell enzyme acting on the exterior viral spike protein or glycoprotein, causing their activation. Their furin-mediated cleavage is required for such protein priming and fusion to the viral host cell membrane and the release of its viral genome into the cytoplasm of its host. Furin is present on the cell surface and in the endosomal compartment, allowing for viral envelope protein cleavage, as in severe acute respiratory syndrome coronavirus 2, SARS-CoV-2 spike protein metabolism, enabling envelope protein fusion. The spike protein detected in the vascular endothelial cell, Golgi apparatus, binds with furin, indicative of the association of furin cleavage, site activation, and vascular cell damage. The furin cleavage site in the SARS-CoV-2 functions as a determinant of COVID-19 pathogenicity, enabling viral triggering of persistent white vascular amyloid clot formation resistant to normal degradative fibrinolysis. The fibrin protein formed from fibrinogen composes an insoluble fibrous mesh in the vasculature, impeding blood flow. Plasmin is an enzyme in blood that attacks fibrin and destroys clots. The clots under consideration are resistant to plasmin since they are not readily broken down and accumulate in vessels, impeding oxygen exchange and blood flow. Fibrinaloids are also referred to as microclots, as observed as a complication of COVID-19 amyloid clot formation. Amyloid is an anomalous insoluble protein fiber formed from clumped together misfolded proteins depositing in tissues and organs, disruptive of the normal function, causative of pathology. Vascular amyloid clots trap proteins such as alpha-2 antiplasmin, which inhibits clot breakdown, and serum amyloid A, which exacerbates the persistence of clots. The serum amyloid A is a liver-produced protein traveling on high-density lipoprotein particles and is a major acute phase reactant and disease biomarker whose level increases up to 1,000-fold during inflammation. Its persistence at high levels results in its accumulation in organs and tissues in the form of amyloid fibrils, causing secondary amyloidosis. Furin is capable of cleaving this protein at the cell surface, and if the virus is internalized within the endosome, it cleaves the protein in this acidic environment. Furin site cleavage separates the S1 and S2 viral subunits during viral entry, releasing the spike protein S1 subunit into the bloodstream, capable of interacting with blood components, including fibrinogen, resulting in amyloid-rich clot formation. Cells possessing an intact furin cleavage site release higher levels of S1 subunit, triggering the very strong immune response, causative of pathological white vessel clot formation. The furin cleavage site enables SARS-CoV-2 to be processed not only by furin, but some other proteases, including plasmin and elastin, playing a role in the pathological vascular complications in acute and long coronavirus disease under consideration in this presentation. This cell surface level modification exposes the furin peptide to elaborate its specific effect. Furin performs as a molecular scissor, activating the viral outer surface during exocytotic maturation and endocytotic entry, allowing for genomic escape from the viral particle instead of its release into same. Furin-mediated processing of viral proteins can occur at several steps of the viral replication cycle. Furin processing induces a structural host rearrangement that allows binding to a secondary receptor and subsequent receptor-mediated endocytosis into the host cell. Furin is substantially present in the respiratory tract and vascular system of human beings. The SARS-CoV-2 spike protein furin cleavage site is an efficient virulence factor that enhances viral entry, syncytia formation consisting of cell-to-cell fusion, as well as cell-to-cell spread, enabling infection, driving the severe pro-inflammatory response and coagulation pathology associated with COVID-19, essential to white vascular amyloid clot disease progression. However, it does not exist as a direct factor in the mechanism of human blood clotting. The furin cleavage site has been referred to as a gain-of-function element, enabling the ability of a virus to trigger the persistent pathogenic vascular amyloid microclots by means of breaking the spike protein in the bloodstream, inducing the unusual clotting described in this presentation. It allows for a virus to be enzymatically cleaved in human tissues, enhancing viral entry, transmissibility, and pathogenicity. Maturation of the cellular protease furin. As a result, furin can be shed into the extracellular space as an active enzyme. Furin cleaves and activates a variety of mammalian, viral, and bacterial substrates. The serine protease furin is essential for proper activation of a variety of cellular precursor proteins. As a consequence, aberrant expression or activity of furin may result in a variety of disorders. Left panel. Furthermore, numerous viruses exploit furin for the activation of the glycoproteins central panel, and several bacterial exotoxins are activated by furin-mediated cleavage right panel. SARS-CoV-2 entry into endothelial cells and early endothelial activation. New presentations coming soon. Like, share, subscribe, and ring the bell for notifications. Thank you to current and new subscribers.